The overall goal of this procedure is to generate bona fide induced trophoblast stem cells, or iTSCs, from murine fibroblasts.
This method can help answer key questions in the stem cell field regarding the nature of the barrier separating the embryonic from the extraembryonic lineage.
The main advantage of this technique is that the transgene expression is only activated transiently, facilitating the selection of stably converted cells.
In a biosafety Level 2 laboratory, using the appropriate personal protective equipment, cover 5 100 mm dishes with 5 ml of poly-L-lysine solution, gently rocking the plates to evenly distribute the coating solution. Place the dishes into a 37°C incubator for 30 minutes. Then aspirate the poly-L-lysine and rinse the dishes 1 time with PBS. Next, plate 7 × 10⁶ 293T cells per dish in 10 ml of 293T cell medium, swirl the dishes to evenly distribute the cells, and return the dishes to the incubator. The next morning, replace the growth medium with 5 ml of transfection medium. Then treat the cultures with 6 microliters of a 1000x stock of chloroquine solution, and place the plates back into the incubator at 37°C. To prepare the transfection mixture, for each lentiviral vector, add 600 microliters of lentivector DNA solution dropwise into 600 microliters of 2x heaps buffered saline while vortexing, and incubate the transfection mixture at room temperature. After